So it's my pleasure to introduce Chris Motyka. I've been working with Chris for a couple of years now, and I'm just amazed at how much I learn from her every time we talk. And so I'm excited to learn more about hemp propagation and seedling production from Chris. She's got a long background in horticulture, cannabis production, now industrial hemp, and she works at VTC. And also does work with some commercial hemp producers here in Vermont. So Chris, it's all yours. Thank you so much.
So we're going to look at a couple different methods, sexual from seed, look at regular and feminized and asexual cloning. The first thing I wanted to kind of point out is we're talking about starts for resinous or high cannabinoid production. The industrial hemp that's grown for seed oil or fiber is grown generally direct seeded, and it doesn't exclude males. In fact, anything industrial hemp grown for seed knows males. The slide on the left is really interesting to me because it's a seed crop, and you'll see the females are the big beefy green plants, and the males in this slide are the tall, kind of pale inflorescences. And these males, they've set pollen and they're senescing at this point, so they're dying off just to make way for the female plants. And I'm going to refer to resinous hemp, that's the high cannabinoid hemp that we're talking about. So sexual propagation from seed. So one thing that's really important to know is that the life cycle of hemp is new. So they're going to germinate, go through a vegetative cycle and a reproductive cycle in one season. And because it's this an annual strategy for survival, seeds are going to germinate pretty readily. So if you can grow a tomato seed, you can grow a hemp seed. They're pretty easy to germinate. Any methods for starting basic annual plants will work for hemp. And then also the thing that's so important to know about hemp from seed is there's going to be genetic variability within that population. So dioecious, this is important to recognize about the cannabis plant, that the male and female flower reproductive structures are on different plants. Regular seed produced from the pollination of the female from the male is going to be 50-50 male-female. And the resin is highest in unfertilized flowers. This is super important for production of high cannabinoid or resinous hemp. Those seeds will, take away from the resources that need to be going into resin as far as producing that product. And the other issue is if there are seeds in the flower from pollination, resinous hemp that is that you're growing for a flower market, a smokable flower market, is basically rendered very low quality, and it's very hard to sell. So for extraction purposes, it decreases the amount of cannabinoids, CBD in that flower. For smokable flower products, it really decreases the whole quality, and you could have your whole crop rejected for the presence of seeds. And so pollen-bearing plants have to be removed from the crop. Even if you have extracted all the males from the population, there's still the possibility you're going to get hermaphrodites, which are just female plants that develop some male propagation structures on that same plant. This can be a response to stress. Sometimes it's just a genetic propensity of that variety, but what it means is you're going to have to look for male flowers even on female seedlings in the population. And those male flowers might be lower down on the plant. They might not be at the very top of the inflorescence. So it's tricky and needs some attention. So feminized seed. So since we're really looking to eradicate any males from the population, one of the tricks is to have seeds that's feminized. So female plants are treated with hormones. This is usually silver thiosulfate that prevents ethylene production in the plant, which prevents the plant from developing female flowers. Instead, that female plant produces male flowers and pollen, which is just amazing and cool. And that pollen contains no Y chromosomes. So if you use that pollen to pollinate a female plant, the seeds resulting from that will be 99 point whatever % female if done correctly. So pretty cool. So regular versus feminized seed. Regular seed is generally less expensive per seed, but it means that 50% of that population of seed is males. And then the trick is you're going to then have to figure out which of those are males. Pre-flowers emerge on those plants 3 to 4 weeks after germination. So there is a way to sort of tell what are the male and female plants, but it's very, very tricky to do it 100%. And that sexing process can delay the transplant of the plants. Feminized seed are more expensive per seed, so you're generally talking $1 a seed or more, but there's less resources wasted on growing males. A small percentage of those plants may still produce pollen, so you still have to scout for males. So I'm, Kristen can kind of jump in on this one, but some of the issues that we've just talked about that in organic certification of hemp seed and hemp seedlings are just finding sources of organically certified seed and some of the feminization process is not organic at all. So I don't know, Kristen, if you want to jump in at this point.
Yeah, I don't know if anybody has like questions in chat about that. I don't see anything. Yeah, I don't have a lot to add.
Do you, and this has been a question that I've heard a lot about feminized seed using those chemicals not being able to be certified organic at that point.
So the seed, so what we have currently is that the silver nitrate or the sodium thiosulfate would not be able to produce organic crops, and then would be considered like parallel production, but and seeds from the treated mother plants would not be considered organic. However, they may be considered untreated non-GMO seed when the organic seed is not commercially available.
That's great. So you could potentially still produce an organic crop from those seedlings.
Yep, from those seedlings. But the seedlings wouldn't be organic. Does that make sense?
Yep.
Okay.
And if people have questions on that, they can just throw that in the chat and we can dig into that further too.
Absolutely.
So just some details about seed propagation. 1/4 inch, no more than 1/2 inch deep. These seeds will rot if planted too deeply. Fresh seed, of course, 1 seed per pot or cell. These things can grow into monsters, so you don't want to multi-seed. It's fairly easy to seed them by hand, but a vacuum or other mechanical seeder can aid in that. Okay, so containers for seed propagation. As with any production of seed, you want to use clean containers, clean media, you know, to not reuse containers that might contaminate, that have some root pathogens in them. And it would be great if we could have containers that were recyclable. This is an issue with the whole hort and ag use of plastics. But the most common cell tray is 72s or 50s, and that would be 50 or 72 cells in a tray that's approximately 10 by 20 inches. 3 to 4 inch plastic pots are also common containers for growing seedlings. Sometimes peat or fiber pots, though I have when they biodegrade, they dry out so quickly that it's tough for the seedling. So containers, the trick with growing hemp from seed for transplant is that if you grow it in a cell tray, you have to make sure that you are timing things such that when that little seedling is has rooted out in that little cell, that it goes to the field pretty quickly or gets up potted into a larger container. If they stay in those cells for very long at all, they'll become root bound, and that plant can be stunted, and it will not grow out of that if it's too root bound or allowed to dry out excessively in that cell tray. And it's tough to keep those things watered if they're hanging around for very long at all. So some producers, and this is a really successful way to do it, is to just direct seed into a larger container. You have then a longer time to kind of play around with the plants and have issues happen in the field and still have a crop that's going to be really viable and strong at going into transplant. The only trick is putting a small seed, and in this case on the right-hand slide, these are 4-inch containers, but they're 4-inch deep. So it's a pretty good sized volume of soil for a tiny seedling. So you just have to make sure that you are watering in such a way to let the media dry out. If it gets too soggy, they won't germinate, or they'll become overwatered and stunted that way too. So it's a trick. You just have to make sure that watering is pretty good. Okay, seed propagation details continued. Temperature between 70 and 80. They're a pretty forgiving seedling as far as temperature. Humidity, above 50% would be helpful. One of the issues here is this is a photoperiod sensitive plant, so it needs long days to stay vegetative. So if you start too early in the spring, that would be, early May, you will need to do supplemental lighting in a greenhouse. You can start these things pretty easily inside using fluorescent lights. T5s, T8 bulbs are fine. You just need to make sure that they do have enough light and they're located pretty close to the light so they don't stretch. That's another way you can ruin a pretty expensive batch of seeds, is having the light too far away so that the seedlings stretch and then their stems become really too weak to transplant well. Media. So we're so lucky in Vermont. We have a lot of amazing producers of organically certified media, compost-based media that the hemp growers have jumped all over. So we're super lucky. But you can use, there's a lot of different ways that you can do this and a lot of different media that you could use. It just needs to be well aerated. Compost-based, peat moss-based, perlite, cocoa coir are all common additions. And then growing cubes. I'm going to jump into this a little bit. Growing cubes have been kind of a medical cannabis, rec cannabis preference for starting seeds, but they're not great for hemp transplants to the field. So these are the propagation cubes. This is an example of rock wool. And there's also growing cubes that are made of peat, cocoa fiber, or hemp fiber with some kind of polymer, or hydrophilic foam, oasis foam has also been used. I'm not going to say there's no certified options for growing cubes. I'm sure there's some out there, but the vast majority are not certified organic, and many are not biodegradable, like this rock wool. It's not something that you want to be planting into your fields. So just paying attention if you are using grow cubes for starting seeds or clones, just to pick one that's biodegradable at the very least. Okay, moving on to asexual or clonal propagation. The advantage of propagating from clones is control. So as I mentioned before, seedling propagation, sexual propagation, so from seed, is going to result in a population that has a lot of genetic variability. Some breeders are awesome and they've really lowered that range of genetic variability, so you have a really consistent group of plants. Other breeders just haven't got there yet, and so you're going to get a seed batch that has widely varying phenotypes in that field. So vegetative propagation, once you have a plant that you know is compliant, it maybe has a super high percentage of CBD, a terpene profile maybe that is really advantageous to sales, you could have a whole row or a whole field of that exact same plant through clonal propagation, theoretically at least. Disadvantages are, to do that kind of propagation, you need stock plants. Stock plants in cold, in Vermont, that's just an expensive thing to keep going. And the pest and disease management is going to need to be something you pay attention to. Stock plants are notoriously dirty, and that would be an easy way to transmit pest and disease through your, all your clones. So I want to kind of take a look at kind of what we just talked about in terms of the challenges for organic certification here. And I'll just read through this, and Kristen, if you want to jump in. So there's just limited certified organic seedling or clone options, and just paying attention to claims and making sure that any sources really, it's truly certified, and keeping good records is important.
So
you go ahead.
Jump in there. So for the cuttings and clones of the hemp planting stock, those also must be certified organic unless not commercially available.
Yeah. And so it'd be really important to keep any records in your, as to your search for certified organic, to prove that at least you've really looked at, making sure option, of course, that there aren't any organic options. And the rooting hormone for producing clones is also the conventional rooting hormone isn't organic, and aloe powder can be used. I've never used it before. I don't know how commercially effective it is, but that is an organic option. You can jump in, Kristen, if you have anything to add to that too. And I don't know if there's anything in the any questions yet regarding this.
Let's see. And so yeah, the aloe powder is one that I've heard of a bit. I don't know how effective it actually is or not, but it is one that we do allow for that. So somebody has a question in the Q&A about what the aloe powder does. I think it would be the same as the rooting powder.
Mm-hmm.
the rooting hormones.
Yep. I mean, I've gone so far as to order it, and there are certified organic powdered products out there that are easy to purchase. I just haven't, actually tried it yet, but I don't know if anyone has out there.
And then just real quick, Nicholas does have a question about the organic certification on the seeds, whether it's non-GMO. So the seeds would themselves be certified organic. So the regulations state that you have to source organic seeds, and if organic seeds are not commercially available, you could use an untreated non-GMO seed regarding that, and that would be the same for the cuttings and the clones.
Great. Okay, just diving right into production of vegetative clones. There's just a little setup, pretty, but needs more stainless steel and alcohol visible, but there it is. So what we're doing is we're taking a branch off a good quality, healthy mother and turning it from that into the little cutting on the right. And I do a note here, usually want to cut the initial cutting a little bit longer than you're going to actually produce as far as the clone. And you're going to want to pay attention to the nodes. So the nodes are the place where the shoot comes out from the stem, and they are a place where there is more active cellular differentiation. So if hormones are applied to those areas, they more readily produce roots. And we want at least 3 nodes on our cutting. And this cutting to the right here, you can see towards the bottom, you can see where the shoot was cut off. So there's one there, there's one just above it where the stem kind of angles. And then moving up, there's one where the little shoot is coming out, the one above that, the one above that. And so there's 1, 2, 3, 4, 5, 6 nodes on this example cutting. So this is the cutting that I would stick. Just note that this is stylistic, doesn't have to look exactly like this. A lot of people like to include more shoots. Some people like to cut some pretty large cuttings. Some people really like to trim them down. But in general, 3 to 5 nodes. The stem tissues should be firm enough so that it can be actually stuck into the media without bending. If it bends or it's floppy, it won't root. Use a very sharp sterile blade, cut at an angle through a node, and then apply hormone to the base. Now there's all sorts of hormones, again, like the aloe powder we were talking. There's other powders, non-organic powders. There's gels, there's liquids. They all seem to work pretty well. People have their preferences. And then you want to stick that cutting into the media approximately 3/4 of an inch. The propagation cubes are already pre-dibbled. They already have a hole in them. If you're making your own media, you want to make a hole first before sliding this in so it doesn't just scrape all the hormone off. And the other thing about this cutting is this cutting has been docked. That is, the leaf surface has been trimmed so that there's less transpiration surface. That has nothing to really do with the success of the cutting. It's all in the aftercare. So I just do want to point out that this is more of a cannabis, like a rec or a med commercial cutting look, and there's differences that necessarily have to do with, the final success. Okay, so production of vegetative clones, the media should be well drained. This is super important, more so for clones than for seedlings. So you can take a soilless mix, and in this, the media, we have issues with the hormone, but the media, we have lots of options, organic options. You don't have to use a propagation cube. You can produce your own soilless mix for cloning. And you know what I've done is just take a really good organic compost-based mix and add organic perlite to that mix to make it really, really crunchy and aerated. And that's worked great. So the other difference between a propagation cube and using loose media for cloning is the propagation cube, when those plants, those clones start to root out, they can root just very minimally, and it's still held together in the cube, and you can transplant that. With loose media, you really need to wait for another 5 days or so before the clone is completely rooted out, such that the cube will hold together, so you don't have a lot of root disturbance. But this example picture here is clones produced in loose media, and in this media, in this case, was Vermont Compost Company's Fort V with added perlite. So those are some nice looking clones. They rooted out really well in that. So, and also you don't want to overwater clones, just like seedlings. Aftercare, this is absolutely crucial in the production of clones. They need bright, they don't need super intense light, but bright light, high humidity. A grow room would be ideal, where the relative humidity is above 70. Then you wouldn't necessarily have to use domes. In this example, domes are being used because the ambient humidity is lower than 70%. So domes can really help to increase the humidity around those clones initially. But you can see the condensation on those domes, also an environment that favors botrytis. So you really have to just be careful that you're burping those domes, maybe wiping those domes so that there's not moisture in contact with the leaf surface, you know, continually for days. Good sanitation is key here. Always use clean tools, clean containers, clean out those domes if you're using domes. Domes are really a double-edged sword. So having a propagation chamber, just a small room where you can have humidifiers in there to get the RH above 70 would be ideal. So first week, the humidity should be 70 to 90, at least 70 to 90%. Temperature around 70 to 80. Some people use bottom heat to speed things up. And then you're going to gently lower the humidity over the next couple of weeks to 50 to 70%. Every single environment is different, so it's really, you know, you got to work with your environment, and what works in one environment isn't going to necessarily work in another. So you always have to kind of experiment. If the plants start to wilt, that is, the transpiration is basically outstripping the plant's ability with its new roots to compensate for that, then just increase the humidity, either with a dome or just increasing the humidity in your chamber for a few more days to try to coax them along. Okay, so that's a lot of resources to have in production of your own clones, and even greenhouse, seedlings require a greenhouse or some kind of a place to produce them. So it might be advantageous to purchase seedlings or clones. The challenges are that you just need to know your source, and your seedlings or clones are only as clean as that grower. So you could really bring in pests or diseases very easily on plants. And then ensuring compliance is going to be key in buying clones or seedlings and buying plants. Plants really are the way you're going to bring in pests and disease in an otherwise clean environment. So just if you do buy them in, make sure that they're quarantined and really checked and dipped possibly before introducing them to your other plants. Okay, so this is something that I've just been super curious about and playing around with, clones versus seedlings in the field. So in this a slide. This is, you know, kind of a rough field, but what I've done here is I took some clones of a variety that I had seed for, and I also planted some seeds. So the seed is on the right and the clones are on the left. The seed population is going to have a variety of different genetic individuals, different phenotypes. Of course, I just chose one to clone, so they're all going to be one phenotype. But the plants, the little row on the left are, you can see kind of the real consistency of the look of those plants. But anyway, it was just really interesting. The clones were smaller. I don't know if that's because of that phenotype. I've grown some monster clones, so it's just I haven't compared them to what that same plant would be as a seedling. So it's kind of hard to know if they're all smaller or if, you know, more research needs to be done. And the yield comparison, I thought that was really weigh this out, but just looking at the plants, I'm not, you know, it would just be interesting to find out what the yield would be between a, you know, relatively similar genetics clone versus seedling. But anyway, fun project. Okay, so you can tell clones from seedlings in the field. If you didn't know any better, you could crawl around and probably tell the difference, tell what that original stock was, whether it was a clone or a seedling, just by looking at branch orientation. So seedlings are strongly opposite, super strongly opposite. And as they get older, as you go towards the top of the plant, that you start to get sub-opposite branches, and that opposite tendency kind of falls apart. Clones are taken from much more mature tissue from those mother plants, and it's lost that real super strong opposite orientation. This picture of a clone structure is a plant that has been pinched any number of times. I think it was an old mother, so it's not necessarily representative of a clone that you just took and didn't, you know, pinch back a lot, but still that the branch orientation is definitely different between seedlings and clones. And then roots are different between seedlings and clones. So seedling roots generally have a tap root. So they have a primary and then secondary roots. Clones, you're just coaxing secondary roots from that node area. So they don't have a tap root. And this can really affect their stability. So this was just a seedling that looked, a clone that looked very, very floppy in the pot. So I sacrificed it and root washed it, and there was a wound on the clone. It had compartmentalized, but it meant that there was no roots on that side. So if this had been transplanted to the field, this thing would just be flopping around. So any root issues in clones, in cloning and coaxing those roots are going to haunt that plant in the field. And then other things, I've just kind of got some slides on that I was curious about. So you can really see just those secondary roots, that fine fibrous root system on the clone on the left, and the seedling on the right. I root washed this because of the root bound nature. I really wanted to see what, you know, became of that root system, but you can really see the larger primary roots and then the secondary roots, and those primary roots they're just not as common in clones. The other thing I wanted to point out about this slide on the right is that plant was started in a fiber pot, a peat pot, and it just wound around the peat pot and then just never really got out. So it just speaks to getting plants out into the field before they become root bound in the pot, because again, those things will haunt them and will decrease their potential later on. And that's another issue that we saw with those circling roots just kept circling and can girdle each other and girdle the stem. Okay, so hardening off plants, we've got to the point where we have either seedlings or clones, and they are ready to go out to the field. You want to start to acclimate them. If you have a greenhouse with roll-up sides, this is easy. You just roll up the sides, roll them down when it's really harsh weather, and keep rolling them up for longer and getting those seedlings or clones acclimated. If you grew inside, you can pop up some little cold frame. And I just set this up with row cover as the protector when nights got a little cold or if it got too windy. But anyway, 5 to 7 days before you transplant, and then they really will just take off without any slowdown. And one thing that I really feel is important is to try new methods. This industry is so new that every producer is kind of on their own learning curve, experimenting with different techniques, creating their own tools and systems. So be sure you keep experimenting. You should set up some kind of little experiment every year, I think, in the field to just further your learning. Using old seed or stock plants as guinea pigs to, evaluate techniques is fun, and always keeping records so that you don't have to make the same mistakes twice. Okay, so I wanted to wrap up some of the source material guidelines in this slide. So finding good genetics is just tricky. You want to look for material from compliant states or material locally. Definitely ask other growers what they're using. That speaks volumes for the potential of success if other people are growing that variety in Vermont and doing it successfully. Make sure you get those COAs, those analytics on those varieties. One thing to note though, on a seed variety, so the COA is going to be a representative, basically an average of those individuals. In most seed populations, you might have individuals that are outliers and might not be compliant, but on average, the whole population is compliant. Just those analytics, those COAs are really important, but you're still going to have potentially non-compliant individuals in a population that you need to be aware of. This is, I think, especially important for growing smokable flower, because you might have product that, where there's just, or selling smokable flower, because one whole bud could be non-compliant right next to a bud that is compliant. And I think that anyway, it's just something to think about. Early flowering genetics are beneficial, so always be looking for source material for the earliest maturing source material in Vermont. The longer it takes to mature, the more at risk you are of losing a crop. So this year there was a hard frost in late September, and whole beautiful, beautiful fields were absolutely rendered unsaleable overnight. So really heartbreaking. So early is better. Inspect plant material for that pest and disease. I already mentioned that. That's absolutely crucial. Inspecting seeds for consistency and debris is also really important. So your seed lots, you could have viruses in your seeds. If there's debris, that debris could have fungal spores in it. So just making sure everything's really clean. And especially for certified organic, keeping records on all your sources, on your source searches, and on all the sources that you're using. And then the future of hemp production. So I'm still hearing noise about tissue culture for, at least for stock plants, in the production of clones. Wholesale production of clones and seedlings is zooping right along. A lot of established horticulture businesses have jumped into that game. Better genetics, they're coming all the time, and we have a lot of room to grow there, but people are, you know, the genetics are improving all the time. And more certified organic options, I think, are going to have to become available, and that's coming. Okay.
Thank you, Chris. That was great. Lots of good information.